Thank you for joining today's webinar, What We Know About NanoEHS Environment. Today's webinar will feature experts from diverse disciplines to share their perspectives on key findings on the effects of ENMs in the environment. I'm Elijah Peterson, research scientist with the National Institute of Standards and Technology, also known as NIST, and the moderator for today's webinar. The 2021 NanoEHS webinar series will focus on sharing what we know about the environmental, health, and safety aspects of engineered nanomaterials. This webinar will feature experts from diverse disciplines to share their perspectives on key findings on the effects of ENMs in the environment. Before introducing our excellent panel of speakers, I want to mention that the NanoEHS webinar series is an important platform for agencies participating in the National Nanotechnology Initiative, the NNI, to share information on NanoEHS research progress and findings. Throughout the series, experts will share the big take-home EHS messages with the broader nanotechnology community and highlight the NNI's role in answering those questions. We have set aside time for your questions to the panel. You can type your questions into the Q&A box. We'll try to get through as many of the questions as we can. I look forward to a lively discussion. Now I will do a brief introduction to the speakers. First is Jorge Hardea-Tereste. He is the Richard Dudley Professor of Environmental Science and Engineering and Professor of Chemistry at the University of Texas, El Paso, UTEP. Professor Hardea-Tereste has led UTEP's collaboration in 3 multi-institution research centers on nanotechnology. His research has focused on developing applications for nanotechnology in agriculture, water treatment, and environmental remediation. Second is Dr. Patricia Holden. She is a professor of environmental microbiology at the University of California, Santa Barbara. Her research background is in civil and environmental engineering and soil microbiology, investigating bacteria as both agents of environmental remediation and agents of environmental degradation. Her current focus includes interaction of engineered nanoparticles with bacterial communities in the environment. A third is Greg Lowry. He is the Walter J. Blanco, a senior professor of civil and environmental engineering at Carnegie Mellon University and deputy director of the NSF and EPA-funded Center for Environmental Implications of Nanotechnology, SAINT. His research focuses on applications of environmental geochemistry, nanomaterial chemistry, and environmental nanotechnology for sustainable agriculture and groundwater remediation, and robotics for autonomous soil sampling and contaminated site characterization. A final comment before I turn it over to the panel with Jorge. I hope you will join us for the other NanoEHS webinars in the series. More information on all the NNI public webinars can be found on nano.gov. You can also follow us on Twitter at NNI Nano News. And with that, I'll pass it over to Jorge.
Okay, good morning, good afternoon, everybody. I'm going to talk to you today about my journey into the applications and implications of nanotechnology in agriculture. My outline is going to have 3 parts. In the first part, I'm going to talk to you about the investigation of the effects of metal oxide-based nanoparticles on crops. Part 2, I'm going to try to answer a question that will we achieve food security via nanotechnology? And in my last part, part 3, I'm going to give you my overall conclusion, my own message. Okay, can you please go to the second slide? Hello?
Yes, this is the second slide. Do you want me to advance one more?
Oh, no, okay. Thank you. So the over past, I would say after my PhD, I've been working with plants and water. Those are my key areas. And on plants, I've been investigating at the beginning of my career toxicity of metals. And then during the past 20 years, I've been working on the effects, on the implications of silicon oxide, TiO2, zinc oxide, and copper-based nanoparticles in plants. These studies have taken a lot of time. For example, the study we are doing now on sugarcane still is not concluded after 6 years of work. We've been studying plant biochemistry, plant nutrition, plant genotoxicity. But today, due to time restrictions, I'm gonna just mention a summary of our work on nanoparticle biotransformation speciation using synchrotron techniques. Next, please. Before I start talking about that, since the students do all the work, I would like to highlight and thank my students. In this picture, you can see 15 of my former PhD students. Now all of them are PhDs. And I like to acknowledge funding for these students through the UCENIN. Next, please. Also, I wanted to highlight my former PhD student, Dr. Nubia Suber-Samena. She was a peer. She was highlighted on page 35 of the NNI supplement to the President 2019 budget request. Next, please. So in part one, I'm going to try to describe what we've done for many years using a very powerful technique based on synchrotron techniques. Basically, it's high energy X-rays that you can investigate the chemistry and the speciation inside of the plants, which is it's an incredible technique. And in this slide, I'm showing you the studies that we've done on zinc nanoparticles in soybean. Soybean is one of the major crops in the world, and we have worked with this crop for many years. So in this slide to your right, I'm showing you micro XRF image of a stem of soybean, and what you are seeing is a tricolor map where zinc is represented by the red color, and you can see around the stem, you can see the zinc distributed around the plant in the xylem and phloem. Next, please. And then we go and zoom in into the soybean pod, and you may not see this if you are not dark enough, but the zinc is already in the pod and in the red. In red color. Next, please. So we studied every part after the treatment in soybean and zinc, and our conclusion, I apologize to some of you that may not work in this technique, but you can see zinc spectra, which is basically trying to find to compare with model compounds what happened inside. So our conclusion is that the zinc oxide nanoparticles are no longer in the upper part of the plant appearing as the nanoparticles, but are like zinc plus 2 ions. Therefore, the nanoparticle is no longer going into the food transfer, into the food chain. Next, please. In this slide, I'm showing you now what happens when you do a study on silicon oxide nanoparticles in soybean. And then I'm showing you again a synchrotron micro XRF images of the pod and also of the nodule. And we do what we call linear combination analysis to try to find out what happened to the silicon oxide. And basically it's looking that very similar, so that nothing has happened in this part. Next, please. And then again, similar to with the zinc nanoparticles, we study all of them, different parts in the plant, and we concluded that the silicon oxide nanoparticles was not biotransformed. So it stay as silicon oxide, and it's gonna go as silicon oxide through the food chain, differently of our results that we found on zinc oxide. Next, please. So in our study with another major crop, which is cucumber, and we use TiO2 nanoparticles. I'm showing you here what happened to the uptake of TiO2 in the root and in the leaf. And this study, we started using TiO2. It's a composition of two different TiO2, 80% anatase and 20% rutile. And it stays like it is, like 80% anatase and 20% rutile in the root. However, in the leaf and trichomes, the TiO2 went up into the cucumber as a rutile form, in a rutile form, not as anatase. Next, please. And then when we went all the way to the fruit, and I'm highlighting this beautiful picture in here, also synchrotron, that was done in France in the SRF. I would say one of the largest synchrotrons in the world. And we show that from the root to fruit, there is translocation of TiO2 all the way. Next, please. So another issue that we wanted to study, and we did this on copper oxide nanoparticles in letters, is that what happened to a nanoparticle that you put it in the environment, in the soil? If you do the experiment as soon as you put it, that's called unweather. But if you let it stay for many days or months, it's called weathering. What happens to the nanoparticle by the environment? So we did this study with letters, and you can see in here that again is a tricolor map, and the copper nanoparticles, or the copper signal, is in the red color. And we concluded in here, in summary, that when you use microscience studies, that the unweathered copper nanoparticles mostly remain as copper-2, the same nanoparticles. However, when they are exposed for many days, when they are weathered, we show a complete different chemistry, different transformation, because the copper-2 oxide goes to copper-1 sulfur and copper oxide complexes. Therefore, weathering has a big effect. Next, please. Also, now I'm going to refer to part 2. In my group, after many years of work, we had found that nanoparticles, in this case, like cerium oxide nanoparticles, can be used as nanofertilizers. Like I'm showing here, increased plant yield. Like in barley, for example, we found that there is a 50% increase in plant yield, and in wheat, 38% increase. These get us into the application, meaning that we can use nanoparticles or nanomaterials as fertilizers. Next, please. Based on this, my good colleague and friend, Dr. Jason White, from the director of Connecticut Agricultural Experiment Station, and I wrote a paper in National Nanotechnology in 2018. The paper is titled Achieving Food Security Through the Very Small. Next, please. The editor of the journal commented on nanotechnology in agriculture that it is an opportunity not to be spoiled. Why is this? Next, please. Now we have on planet Earth 7.8 billion people living. Next, please. And it's projected that the world population is going to be 9.8 billion in 2050. So how are we going to feed the world in 2050? We're going to need more food, more biomass, more biofuels. Next, please. So we're going to need investment in sustainable agricultural production, capacity, and rural development. And also, very importantly, we are going to need to promote technology change and productivity, for example, using nanotechnology. Next, please. The problem we have, next, is that the current pesticides and fertilizers are not efficient. The nitrogen use efficiency is 30%, and the pesticides, 90% of the pesticides run off into the environment, thus creating pollution. Next. We think that engineered nanoparticles have unique physical chemical properties compared to the bulk nanoparticles. And as a chemical engineer, as a chemist, environmental engineer, I can tell you that we can manipulate the nanoparticles to create nanoemulsions. And therefore, we can provide a time control and target a specific delivery system. Next, please. And as shown in this slide, nanoparticles can be delivered slowly, slow release, and very efficient. Next. So we conclude this by telling you what we think about the future of nano-enabled agriculture. So if we use this in the future, we're going to have better food security. We're going to have sensors to determine freshness of the fruit. We're going to be able to use a smart release of agrochemicals. We're going to have controlled pests, suppress plant disease. And also, we are going to have a better efficiency. The stress tolerance is going to be more efficient by controlling the delivery of genetic material. Therefore, we will have an efficient use of water and energy and less environmental impact. Next, please. Now, I'm going to give you my take-home message, my overall conclusions in this slide. We just recently published this paper, and in this paper, we selected only rice plants. What are the overall effects in rice plants according to different types of engineered nanoparticles? And as you can see here, we look into so many of them, and we found a positive effect. There are some negative effects, but mostly we found some positive effects. Therefore, my conclusion is going to be in 4 points of where to move based on this study and all my plus 20 years of work. The first one, so my conclusion is the first is studies on the environmental behavior of engineered nanoparticles with soil and their weathering properties need to show realistic environmental conditions. Some people are doing studies at very high concentrations that are not the environmental concentrations that are present. Next, please. Because the interaction of plants and nanoparticles is very complex, we need to use novel techniques and instrumentation as the one I showed you, because we require them, and we require to improve the detection and measurement and of the potential effects. Next, please. We need to understand the mechanisms of interaction between nanoparticles and crops. We cannot just throw, I've been contacted by several consultants and companies, and they are putting nanoparticles, but without doing the studies. We need to understand the mechanisms of interaction, what is happening. Next, please. And the last message I have is that the efficacy of between engineering nanoparticles and as potential nanofertilizer compared to the corresponding traditional fertilizer is still unknown. There is still much to do on research in this area. Next, please. I just like to acknowledge my students. I mentor 42 PhDs, students that are working everywhere, including federal agencies, and I thank them for all the work. Next, please. Also, I'd like to thank the support through the years of University of Texas System, the NSF EPA and USDA, NSF of our new ERC, and all the synchrotrons in the world. I'm mentioning here Stanford Synchrotron, the Advanced Light Source in Berkeley, the European Synchrotron, but also my NIH funding, also my research group and many collaborators around the world, including the next speaker, Dr. Trish Alden and Greg Lowry. And thank you for your attention.
Great, thank you, Jorge. So Elijah, you want me just to jump into it?
Go for it.
Okay, great. Yeah, so Jorge, I appreciate the sort of forward-looking and new applications of nano and the promise that it holds, in particular in agriculture. I'm going to take a little bit more retrospective look sort of back at the things that we've been doing in the centers and even a little bit prior to that, and kind of look at what are some of the questions that we had in 2008 that we've answered, and what are some of the open questions that are remaining. But I do want to start by acknowledging a number of things. I mean, the funding clearly was important to get these things going, and the centers, the two centers, Saint and UC CEIN, were really nice centers to pull together the information and work on these big interdisciplinary teams, and you'll see that makes a big difference in how we did our business. But I do want to say something. So Barb Karn, Nora Savage, Mike Rocco, all played a really big role in this if you go back to the beginning and sort of promoting the applications and then getting into the implications and trying to develop them simultaneously, which I think is an important message that we've all learned in the process here. And Hongde Chen, of course, is pushing this and doing a lot of the agriculture and nano and agriculture work. I think that's important. Also, you know, what I'm going to show you today is a center perspective. So all the faculty that I've had the chance to work with over the years on these projects that were both in and outside of Saint have all been fantastic. And the number of students that we've put through these centers and you know, through this interdisciplinary research machine, I think speaks volumes to its success. Okay, let's go ahead and go to the next slide, please. Oh, maybe click it again. I'm not sure why it looks like. I just keep clicking until all that stuff's up. One more maybe. Okay, that's probably good. All right, so this was kind of how we framed our center, and this is how we operated in the beginning. And really, if you look back at the research strategy documentation, these were many of the questions that we had. And really, we wanted to know, can we predict the risks, right? And I put benefits in there too, because as Jorge just talked about, for plants in particular, there's lots of benefits to be looking at. And it's really about the balance between the two. But we started thinking, can we figure out what the risks are of these materials? And, you know, answering the questions like, who's exposed to them? What are they exposed to? For how long are they exposed? And then what are the consequences of those? Are there any toxicity concerns, both acute and chronic concerns? And then, of course, I'm going to focus today on the transport and fate piece of this. And Trish will talk a little bit more about the ecosystem ecology piece of this. But we ask questions like, how do they travel around? How do the properties affect this? How do they get transformed? What do they transform into? Are they bioavailable or not? And we really wanted to know whether we could predict all of these processes from the nanomaterial properties themselves, because if we could, then we could design them to be safe. And then, of course, if we know the answers to all these, maybe we could regulate them better. Next slide, please. All right, so the first lesson that we learned, all of us learned, I think, was that the environmental complexity can't be ignored. You know, if you look at the left of this slide, what we wanted to say is, from the nanomaterial properties, can we predict all the effects that will be out there? And it turns out that you really can't do that. The nanomaterial itself is defined by not just the materials properties, but also by the environmental system that it's in. You know, what is the redox state? What is the pH? What are the organisms that are present? Those are all really important determinants of how they transform, what byproducts get transformed, and who's exposed to what. And only through combining both the material properties and the environmental properties can you determine the effects. And really, this led to a lot of mesocosm work, both in our center and in France and in the UCCEIN, where we looked at all these things in a very complex environment to understand how they would behave. Okay, next slide, please. Okay, so another lesson that we learned was that nanomaterials are unique. I mean, one of the things we went into this was asking, are there unique properties of nanomaterials? Are there unique outcomes of nanomaterials? And it turns out they really are unique. but not in the way we first envisioned. You know, we first envisioned this unique reactivity, that you're going to get these very special reactions that can occur. And that can occur on very, very small particles, but on most of the particles in commerce and the things that we studied, they weren't unique in the reactions they performed, but they were unique because they had a very high surface area, so they performed a lot of those reactions per mass. Okay, so that is a unique feature of these materials, is they're highly reactive. Then the second uniqueness, I would say, is both a spatial and temporal uniqueness. So they move in the environment in ways that are different than an ionic form of a material would move, or that a bulk particle would move, right? They have access and distribution to smaller places. Cellular uptake pathways might be different because they're small. So I call that spatial uniqueness, and just how they're distributed. The other unique thing is that they do have temporal uniqueness. So, you know, they're metastable by I don't know if by definition, but they are metastable materials, these nanomaterials. They are going to react and degrade and change form over time. And that provides opportunities almost for free for things like sustained release, but it also affects persistence. And it's different than ionic forms of materials or bulk forms of materials. So they are unique, but again, in ways that are spatially unique and temporally unique. Next slide, please. All right, and then another thing that we spend a lot of time talking about and thinking about is, you know, the focus is on engineered nanomaterials, but natural nanomaterials are everywhere. And this, you know, Mike Coachella sort of spearheaded this at Virginia Tech, in looking carefully at what are the budgets of nanomaterials in the environment. And if you look closely, I mean, these numbers in this chart are in teragrams per year, which is basically thousands of tons. It's a lot of material, much more than we are producing of an engineered way. And one of the things that we wanted to focus on and are still working on is how are the engineered ones truly different from natural nanomaterials? And are the processes different? Are their reactivities different? And I think this is an interesting question. And we made a lot of progress here in showing that many things are similar, but there are some differences. Let's see. Yep, next slide, please. Okay, so one of the other things that came out of this center effort, and I think really needed a center to be able to do this, was that we now have models to predict the concentrations of these materials in the environment. So this is one of Arturo Keller's work from the UCCEIN. And you know, a number of people around the world have been working on these types of models. But essentially knowing something about the sources, or assuming something about the sources, you know, where the material is at end of life, and then what are the releases, you put those into an environmental model, and you can look at where they move around and distribute, and you can calculate concentrations. But if you look carefully at those concentrations on the right, for many of these materials, these engineered materials, we're looking at micrograms per kilograms or less. So part per billion sort of levels out there or less. A few are higher, so TiO2 is probably one of the highest ones, I think, on this chart, and it's about 100 ppm in a sediment is what they estimate the concentrations to be. So the numbers are pretty low in terms of what we expect and for environmental concentrations of these materials. Now, keep in mind, a lot of assumptions go into these models, and maybe some of them are justified, maybe some of them are not. But we really need to be able to go out in the environment and sample these things carefully and understand what those concentrations really are to validate these models. Next slide, please. Okay, our center and people around the world have spent a lot of time characterizing the transformations of these nanomaterials, both carbon materials and metals and metal oxides. And there's a couple of nice reviews that came out recently in Environmental Science Nano that sort of talk about or summarize what we know about these various transformation processes. My own work looked a lot at transformations. Jorge just talked a bunch about transformations. So a lot of the tools we've developed have been aimed at looking at how things change and transform. And I think we've done a pretty good job of categorizing the behaviors of a subset of nanomaterials. Some of the most commonly studied ones: silver, copper, copper oxide, iron oxides, these kinds of things. There are many other types of materials that maybe we haven't characterized, but a lot of what we learned here, I think, can port over to these other types of materials. So bottom line is we've done a pretty good job of elucidating the transformations, and we can kind of understand if this material went into this environment, this is what we'd expect to see and what we'd find. We've also understood a little bit more about the implications of these transformations on toxicity and reactivity. So for example, silver, if it sulfidizes, it generally lowers its toxicity in certain environments. Okay, next slide, please. And then the transformations were worked on for a long time, and then models were worked on. And more recently, people have been, and a lot of this is coming out of Europe actually, with a project called Nanophase. We've been able to understand how the transformations go affect the models and affect the transport and where these things go in the environment. So we can do a little bit better job of understanding concentrations, exposure concentrations, and then what's there. We know what the form is that people might be exposed to. So I think this is a pretty important step for us. You know, one of the questions I had last year or the year before at the Nanophase meeting were, we've got these models now, we can figure out where things go, we can put most of the processes in, but are these models sufficient for regulatory decision making? Could a regulator take this model and say, okay, plug it in, predict the concentration in the environment, and then we can make a decision about how much we can make or how much you could release, what your permit would allow you to release, et cetera. So I'm not sure that we're there yet. I would love to have that discussion as part of the discussion of this whole event. Next slide, please. I'm almost done. Another thing that's really come out of the last 10 or 15 years is that we've gotten a lot better at characterizing the nanomaterials in the different environments. And we've also done a good job, I think, of separating the types of properties. So there's both intrinsic and extrinsic properties. And we define intrinsic property by a property of the material that is independent of the medium that it's suspended in. You know, particle size distribution, for example, that's something that you can get with TEM. There's automated methods to do that now. And then we separate those kinds of properties, which we're quite good at measuring, from medium dependent properties. So when you put something into a different medium, it might have a different value. Zeta potential is a great example of this, where the value that you get really depends on what else is in that system. But we now appreciate that a lot better as a community, and it's done a lot of service to us to be able to compare results across different studies. You know, there are some issues here, like the EU definition of a nanomaterial with 50% of the particles being, by number, being great or less than 100 nanometers. That's still not that easy to do systematically in a batch of materials. There's batch to batch variability, et cetera. So there's some work that still needs to be done here, but I think we've made huge progress. One of the things we have made a lot of progress on is thinking about how proteins and other macromolecules attach to particles and what influence that has on their behavior, and what proteins are absorbed, and how we can actually pull them off and measure what proteins are there with different mass spec methods. So there's been a lot of advancement in the way we characterize our materials, knowing that this fundamental characterization in the medium of interest really has a huge impact on how they behave and what impacts they may have. Next slide, please. Oh, you can click again. I think there might be some more stuff here. Yeah. Oh, yeah. Click through all of this. Sorry, I forgot there was a bunch of visualization in here. All right, that's good. Okay, so other new techniques have come online. So there's this thing called single particle ICP TOF MS, which is a time of flight MS. So single particle ICP allowed us to start to count particles. And in that middle plot there, you can see all those spikes. They represent a particle. The time of flight mass spec now lets us look at not just the particle events, but it gives us a mass spectrum of all the elements in each individual particle. And we can look at thousands of particles per minute. So basically, the point here is that we've really appreciated the heterogeneity of these systems. Not all nanoparticles in a batch are exactly the same. They have impurities. They might have different sizes and shapes. But here we can look at chemical impurities on a particle basis. So this is a pretty neat advancement, I think. And it has applications in air pollution and particles and air and how heterogeneous they are and which ones are more toxic and which ones are less toxic. I think there's just a number of things that this TOF-MS is going to be able to do for us. And it's the reason it became popular was the need to characterize these materials in environmental media. All right, next slide, please. All right, so this one might be controversial, but we'll talk about it. As of around 2018, there start to be some discussion that the risks from nanomaterials might in fact be quite low. The arguments are the toxicity of a lot of nanomaterials is generally low if you compare them to things that we know to be very toxic, like DDT and these kinds of things. And the predicted exposure concentrations are quite low. So you saw Arturo's model suggest that they're low. So if you have low concentrations and you need high concentrations to be toxic, maybe the risks aren't so high. And there's a couple papers here in ES&T or perspectives that sort of describe that. They're worth a read. I think that that's generally true. I mean, things aren't toxic just because they're small. You have to worry about what's actually it's made of, and you have to worry about where it goes. But there are some scenarios where maybe we need to do a little bit more work, sort of their use in agriculture. There's clearly potential for exposure under those kinds of scenarios, and we might want to think about that. But keep in mind, we don't have to start from scratch, right? We know a lot about particles. We know a lot about toxicity. And we just have to apply them to these various scenarios. Next slide, please. All right, so this is my last slide. There's still a few data gaps that I think remain. So one of the things is about chronic exposures. We know many of these materials will be out in low concentrations and persist. If your ecosystem is exposed to long periods of low doses, what impacts does that have in the long run? Those are hard studies to conduct, and we're not so great at that, or at least we haven't had the time to do those studies. There's still an incomplete picture of nanobio interactions in general. Trish is going to talk a little bit about that, and Jorge talked about the translocation in plants. Being able to deliver something in a plant like drug delivery in a human, we can't do yet. We just don't know enough about how the particles are interacting with the various plant organelles and things to get them where we need them to be. We need to validate our transport models. So we're going to need very accurate measurements in real media at extremely low concentrations. So some more advancement in measurement. Elijah, that's NIST job. Some more advancement there we're going to need. And then, the big question, I think, is all this advancement we've made, can we regulate these nanomaterials now that we know and know how they behave and we know what models are available? And I'll stop there, and hopefully that will spark some discussion. I'll turn it over to Trish.
Good morning. My name is Patricia Holden, and I'm very happy to be here to talk about more of the kind of toxicological aspects following on the great information provided by Greg and by Jorge. And obviously, one thing that I think we can get clearly from their two presentations is that the investment continues. There's deep knowledge that was gained about how to acquire knowledge, and that's the first thing I want to emphasize in my presentation. And then we learned some very long-term tangible facts and about mechanisms and about properties of nanomaterials in the environment. And then the work continues because nanotechnology is mainstream in our economy and continues to offer a lot of promise, solving problems. And there's a deep expertise that can be leveraged for that. So I want to credit the UC CEIN, UC Center for Environmental Implications of Nanotechnology, and also I was involved early on in the EPA STAR program, the NanoSTAR program, and a shout out again to Barbara Karn and Nora Savage for all the support early on in building up this expertise. Next slide, please. So the first thing I want to say is that, I want to look back at what did we do right? So when I look back at the 2011 roadmap for what should we be doing to understand nanotechnology and its implications, and what kind of research should be happening. And in a sense, the train had already left the station. These centers were quite mature. There was quite a lot of expertise. But when I look back at that document now, what I see is that we made some good decisions. That is, there's so many different types of nanomaterials, and at the time, the ones that were being produced in high volume on the market, and the volume here should really say tons per day, they're incorporated into everyday products. You can just see from the short list that common nanomaterials, there's so many of them that we could have selected from to try to understand their environmental implications, their effects on ecological processes and receptors. But one of the things that these centers and the scientific community in general, so it wasn't just limited to the centers, did was to judiciously select nanomaterials that could have the highest probability of exposures in the environment. So that was an important decision that was made early on. And then also the prioritization of ecosystem services. So while we could have, as a scientific community, conducted toxicity studies using standard organisms and standard assays, what the scientific community did really well to make this work and trying to understand the implications of nanomaterials in the environment, what the community did really well was to focus on ecological processes delivered by ecological systems that most impact humanity, most impact society. For example, food production. So Jorge's emphasis in his presentation on plants and their uptake of nanomaterials, very central to what concerns us as human society. Waste treatment. And then also, interestingly, one that we don't often think about is the continuous ongoing many microbial reactions that are catalyzing nutrient cycling, so the return of carbon through the biosphere and nitrogen, et cetera. And so these major ecosystem services, within those, there was a prioritization there. Next slide, please, to judiciously select where should we focus our efforts, which environmental receptors. And then building on what Greg was saying, the other thing that has been done really well, and the reason I emphasize kind of methodology and approach is because this threads through the scientific community's outlooks on how we should be trying to understand emerging nanomaterials in the environment as we move forward as a nano industry. So one of the things that was done in lockstep between the ecologists, ecotoxicologists, and the chemists and material scientists was to say to ourselves, what's being produced out of the manufacturing plant might be very different in terms of its presentation to organisms once that's incorporated into secondary products. So for example, carbon black, a standard of a long-standing material being incorporated into tires, is it the same when it's in tires? Is it the same when it's released? And we recognize that there's a high probability that it's not, and we must understand if it's released, will that be presented to the organism and the environment or affect the ecological process? And if so, that's what we should be studying. We also recognize that as we move through the life cycle of nanomaterials from primary manufacturing through incorporation into products, through release, that in the environment, there's processes that are taking place, as Greg and Jorge described. There's the formation of larger structures, nanomaterials adhering to engineered nanomaterials adhering to natural nanomaterials that Greg described in the environment, forming what we call heterostructures, or adhering to each other, forming homostructures that then differently present themselves to organisms and have a different tendency to be taken up by organism and cause responses at different tiers of biological complexity, from the biomolecular tiers of physiological, genetic, metabolic, reproductive, all the way up through ecosystems. And in keeping this outlook, so this is new, and this was very specific to understanding nanomaterials in the environment from an ecological lens, is of the scientific community paying attention to the fact that the materials were changing in the environment, and therefore that would change their profile of interaction with organisms. And in recognizing that it's the physicochemical conditions of the environment where the nanomaterial will reside, and Greg alluded to this as well, the ionic strength, the pH, the temperature, the oxidation or reduction status, et cetera. That would dictate the form, that would affect the bioavailability, that would affect ultimately the hazard. So these are things that the community, the scientific community, did well. Next slide, please, to be attentive to these issues. The other thing the scientific community did well was to attend to, as I mentioned a minute ago, all levels of biological organization. So on the left-hand side, this is a diagram sort of oriented towards a few terrestrially, so land-based ecosystem services, food production, growing of crops, as Jorge mentioned, showing the soybean here, and waste treatment. So these are important endeavors that human society has, and the field explored concertedly these important endeavors for their potential threats or hazards from nanomaterials as they enter into these systems. But again, it wasn't a matter of looking at standard dose response types assays, but really asking what are the functions that are occurring, ecological functions that are occurring in these systems, and then how can we best study them in an efficient way moving forward. There's so many different types of nanomaterials at the beginning of this scientific endeavor, and now the numbers of different types of nanomaterials continues to increase. We looked at not only understanding for the highest volume materials, the mechanistic relationships between nanomaterials and organisms, but then we use the understanding of those mechanisms to develop screening studies. So how could we rapidly screen nanomaterials that have similar likely interactions with cells based on those nanomaterial properties moving forward, and then elevate from those screening studies those nanomaterials into more complex toxicological testing, mesocosms, looking at entire plants or looking at pilot scale waste treatment systems to understand what is the real potential for effects. So these are more things that the scientific community did uniquely in this space and did very well. Next slide, please. The other thing is that as we looked at, and when Jorge was talking about nanomaterials moving up into plants, we're talking about this phenomenon of bioaccumulation, bioconcentration, and potential for biomagnification. And that's partly what was explored early on in the plant world. And some of the early discoveries were coming out of the CEINT that Greg was referring to, looking at trophic transfer from insects that were chewing on plants that had taken up nanomaterials. There were also microbial trophic transfer studies looking at the potential for trophic transfer as a pinnacle concern in ecotoxicology. And we know about biomagnification. We can think about DDT in the environment, worst case scenario potentially, or mercury, where these chemicals build up in the food chain, higher and higher concentrations, the higher you go up in the food chain. And so the scientific community was attentive to this as a possible concern in nanotoxicology, and not only looked at it in a number of select systems that could be important from an ecosystem services standpoint, but then also said at the through a review process and an international process that was actually led by Elijah Peterson, our moderator here, what are the best practices for future assessment of bioaccumulation, these concerns in ecotox that are not only affecting the dose that the organism is experiencing over long time scales, but also affecting the transport, because as critters, as organisms, as plants accumulate nanomaterials, those plants, those organisms can also be vectors further in the environment. Next slide, please. And as Greg alluded to as well, and Jorge as well, there was a very deep investment in trying to understand mechanistically on the basis of the physicochemical properties of the nanomaterials, how are they affecting cells? This work was led, very brilliantly by Andre Nell within the UCCEIN, looking at human cells, looking at mammalian cells, and using those to advance high throughput screening methods. Our center also looked at this from the standpoint of bacteria, which are the most numerous organisms and undoubtedly the most catalytically important organisms in the environment, because understanding the mechanisms of interactions could allow the scientific community, and this has gone on all over the world. So the entire scientific community looking at nanomaterials has, from a biological standpoint, has investigated mechanisms to some degree, asking how can we relate the interactions of the nanomaterials with cells to properties of the nanomaterials, and then use that to guide design into the future, perhaps designing safe nanomaterials on the basis of avoiding the types of harms that are anchored in these mechanisms of interaction. And so this diagram here really, it's complicated, but I think the message here is that the complexity reflects that the scientific community thought comprehensively about the nature of the interactions, and this required that there was this, interdisciplinary kind of thinking across not only the materials and the organisms, but the threading across those two areas to make a long-term knowledge base available in this field. Next slide, please. And then moving up in biological, so not only understanding mechanisms, but then going back to that whole mindset of ecosystem services and saying, what is the potential that a soybean crop in a field could be affected by nanomaterials to the degree that more fertilizer might be needed, more nitrogenous fertilizer. And nitrogenous fertilizer, the synthetic material, requires a lot of fossil fuel to produce it. So there's a high global warming potential of producing more synthetic fertilizer. Leguminous plants like soybean can naturally recruit dinitrogen from the atmosphere and sort of self-fertilize. And so there was this, again, focus within the community on plants like this and other plants that were providing the food aspect of ecosystem services, and then asking, how might this be disrupted in a way that we should control for, be attentive to, and use as a model system for studying in the future. And the other thing coming out of the CEINT was to look at, in both of these settings, the idea was that the nanomaterials, the vector for them into the environment could be biosolids from wastewater treatment, because disposal to agricultural lands is a commonplace practice in the United States. That could occur in, environments where greenhouse gas production is also affected by soil microorganisms, which was found in this important study out of the CEINT, where it was silver in many different forms, including the nanoform, that affected that. So we were able to look at ecosystem, the potential for ecosystem level effects. Next slide, please. And then moving forward, what happened is the field developed is, the capability, the orientation, the approaches, the methodologies for articulating this orientation to the research that the scientific community really thoughtfully developed. As that matured, then the recruitment of new technologies to continue to study nanomaterials and their interactions with organisms was also happening in lockstep. And so, what that has primarily involved is using what we call omics methods, not only looking at does a nanomaterial interact with a cell, but what sorts of genes are transcribed differentially, and then how does that change protein production, including catalytic proteins, enzymes? How does that change entire metabolic regulation or metabolic systems? And this became very important to look at, because as both Jorge and Greg mentioned, the amount of nanomaterials in the environment may actually be very low for any particular kind. And so when we're trying to understand effects of materials on organisms, we have to be able to have sensitive metrics. And one of the things that the scientific community understood moving forward was that these omics tools were very profound for understanding the sublethal effects and could be the basis for making decisions moving forward. Next slide, please. So I want to wrap up and say, the long-term benefits, one of the things that we said, the scientific community said to itself during this process and it's continuing, is that this was a unique opportunity to develop an understanding of the potential environmental implications of a new technology while that technology was developing. And that there should be opportunities, as Greg mentioned, and to assist with new product authorization, that is from a regulatory standpoint. The methodologies, the approaches, the ideas, the way that the work was focused across the globe, highly transferable to other problems that are very analogous right now: nanoplastics, next generation nanomaterials, even viral particles, thinking about small particles and how they may modify in the environment and affect ecological processes. And also to emphasize here is that, one of the gaps was looking at nanomaterials and mixtures and looking at nanomaterials and mixtures with other substances. That's an area that's an opportunity that with this set of transferable ideas, approaches, and methods could really be exploited, and we could learn a lot more. And then lastly, as Jorge showed with his pictures of his many fantastic students and postdocs, the thing I want to emphasize here is that the trainees that were produced through this investment and continue to be, they're highly interdisciplinary. They understand material science to a degree, inorganic chemistry, organic chemistry, molecular biology, ecology. They understand the societal relevance. They are not narrowly nanoscientists. They comprise a very versatile scientific workforce, and really they're there as a tremendous opportunity moving forward on a lot of different research fronts. Thank you.
Thank you, Greg, Trish, and Jorge, for sharing your perspectives in these excellent presentations. And also thank you to our attendees for being part of today's event. So let's go on to some of the questions. There is one detailed question that Jason, or I guess two now, that Jason White sent that I think would be interesting for us to discuss as a group. One is related to the regulatory approval issues that we did discuss, but then also the societal acceptance dimension. If there is a great product, I'm paraphrasing, but if there's a great product, but then there are concerns that people don't want to eat it because it's contaminated with, quote, nanobots, then that could be really challenging. In some of his discussions, he suggests that's this issue of societal acceptance is as big of an concern as regulatory acceptance. So what would you all say to that?
So Jason and I have talked about this before, and we run into this a lot when we propose the use of nanomaterials in agriculture. One of the approaches that we're taking now is to move away from synthetic type of materials and try to use biopolymers, try to use mineral species, things that are generally regarded as safe as delivery vectors. That helps with the vector part. But if the reality is you're delivering copper or something that could potentially have some toxicity associated with it, one way to think about it is that we've been using copper on agriculture for thousands of years. We've been even using nanoforms of copper. I think there's a lot of data out there, as the question suggests, that we know how to predict and understand the fate of those materials in the environment. There's even work out there about the fate of those materials on the plants themselves, even edible plants like lettuce. So the evidence is there to suggest safe use. We just need to do a better job of communicating that evidence to consumers and presenting it in a way that talks about the risks and the benefits. And in the case here that Jason tried to lay out, the benefits clearly outweigh the risks, you know, doubling soybean production. But that's, people do understand risk, right? People get how to manage their own risks, and they're willing to take risks if they see benefit in it. And we just need to be able to communicate that better. I thought I saw Kara Grieger on the line. She might have something to say about this too. She's doing a lot of work right now trying to understand those perceptions. I'll stop. Hopefully there's others that will chime in.
I think unfortunately the participants, I don't think they're able to speak, but even though some of them may have great expertise on the topics.
Oh, okay.
Yeah, I just like to share my experience in another center that I'm part of, NUT, ERC, that we are producing drinking water. And we have done several surveys, the group of Paul Westerhoff, and again, I echo Greg's comments that we need to let the public know what nanotechnology is. If they understand that we are not, there are no unintended consequences. So if we show them that if, for example, if we are producing drinking water, the drinking water is free of any potential nanoparticles that could be there, the public will understand. But our surveys, our studies have shown that the public still doesn't know much about nanotechnology. And I'm sure Trish can mention about the social study that we did at CNIN. Trish?
Well, the survey, are you referring to the survey, Jorge, the surveys that were performed? Yeah, and there was also societal relevance that was performed by Barbara Hare Hawthorne. She led that in the UC CEIN very brilliantly, and I'm trying to look at, they did surveys with, with many different groups, trying to understand their perspectives on risk, and they had a lot of the same questions that we're talking about here. And so I think those questions still remain. And we get those all the time as nanoscientists. Our neighbors, our friends ask us these questions. Should I wear my sunscreen? What's the benefit of that versus the possible risk to myself and my family and the environment? And we really could come together as a community and do a better job of talking about that. It's probably too much to cover right now, but suffice it to say that, and because there's so many dimensions to it, there's food, there's other personal care products, and then there's just, concern for the environment at large, that where we're using nanomaterials in a lot of products, are they interfering with systems that would require changes, adjustments that can affect our global health. So we can do more on that. We need to.
Thank you. I'll throw out a related question. Back in the early days of NanoEHS, we saw a bunch of work on environmental remediation being a potential application of nanotechnology. Now haven't really seen as much about that lately. And I know, Greg, you're talking about maybe some of the risk trade-offs may not be as severe as we thought earlier on. What do you all see as the potential future for other environmental applications for nanotechnology in addition to agriculture?
So we did a paper last year that came out of a workshop in China on nanomaterials for remediation. There's a lot of opportunities to degrade things that aren't degradable with current technologies, to reach places that are difficult to reach for remediation. So I think that's a great opportunity. Another opportunity that I run across a lot and see as important is resource recovery. So ways to recover nitrate from wastewater, ways to recover phosphorus and potassium and various things. Nano approaches and membrane technologies that rely on nanomaterials for that look like they're going to have some promise moving forward. Fortifying foods, I guess you said other than agriculture, so I'll let that one go. Those are the two that popped into my head. I'll see what the others say.
And also, I just want to comment that following up on our work on remediation, so depending of the nanoparticle or nano engineered product that we are talking about. So if our many years of work in that field of phytoremediation shows that if you have an organic compound, that is toxic, PAHs, that can be degraded, I'm sure it's going to be the same situation in the nanotechnology area. And now if you have a compound like TiO2 that we cannot do that. And I don't want to go to the issue that is going on in Europe now in respect to TiO2, right? So again, if you design a nanomaterial that can be degraded with time, and as Greg mentioned, it's not gonna stay in the environment for a long time. That's the way to do it.
Thank you. And Katrina Werner also chimed in about how it's being used in roadways and concrete mixtures and other types of ways for the built environment. So we'll go to another question in terms of going back to agriculture. Jason White was asking about how concerned do we need to be about worker or grower exposure for the people in the field who are applying some of these nanopesticides or nanofertilizers.
You want this one, Jorge?
Well, again, depending on what, right? If this is going to be something that is going to apply at high concentrations, they need to use the PPE, right? They need to be protected. The case of nanosilver, that it was approved by the EPA to be used in agriculture. Now there was an issue that now we are not allowed to do it. So we have to be, again, we have to have more mechanistic data on the chronic effects, right? And I think in the case of nanosilver, we didn't have. But if we are designing the future pesticides in agriculture, I mean, we have to do studies that those are going to be products that are not going to be toxic.
Yeah, that's what I was going to say too. The protective equipment for applying chemicals and things with particles in them, it exists. We use it, but we also have to do the studies for human exposure to these new materials to make sure that they are going to be adequately protected with the PPE that we have.
Thank you. So another agriculture-related question: when you have a monoplanted crop that is more vulnerable to plant pathogens, how do you modify the nanoplant pathogen treatment in response to plant resistance to the treatment, such as wheat rust?
That's you, Jorge.
I think we can do it, but that's not my field. If Jason is present, I will say, okay, Jason, that's in your field. But that's, yeah.
Yeah, I mean, so my answer to that is there are nano, and we did this paper in Nature Food that came out, I don't know, late last year or something, where we talk about sort of the technology readiness of various nanomaterials for agriculture. And nano carriers for pesticides and various things, and the ability to target those carriers to certain locations in the plant are pretty promising. So if you want to target a very specific pest, and you work with a plant pathologist who understands that pest, understands how it gets in there, understands how the disease manifests, then we can design something to try and get in front of it. But we can't do it alone, right? We need to work in interdisciplinary teams, and we need to have very specific marching orders for that pathogen, and then something could be designed to work there. Now, if there's classes of pathogens that behave the same way, maybe you could come up with something that would work on a whole class of pathogens. But that's happening, right? The agrochemical companies are working on these kinds of things. They don't share very well with the others, right? They don't publish in the papers, but it's definitely research that's going on.
And I just want to mention that, as Greg said, that the companies are working on this. And I mean, I cannot say what companies I am working with, because I am working with some companies on this issue, the big ones in the world. We have several students that work for CNIN. Trish, you remember one, is working for, I cannot say the big names of the companies, right? But most of the agricultural companies have people trained by Greg, by Trish, by us. And I mean, because of the, the patent issue, we cannot really say much about that.
Okay, so I'll take the last question, throw it out. So how do you all see the all this research on NanoEHS and engineered nanomaterials applying to incidental particles such as micro and nanoplastics? Is there, what do you see as some of the ways that what we've done could support understanding the potential risks and environmental fate of those pollutants?
I can start there, because in our group, we definitely have gone in the direction of plastics because there's a lot of student interest in this area. And just to say again, the field has prepared all of us to think about this very high profile issue. And of course, many of us know that the biggest challenge right now is trying to quantify nanoplastics in the environment. So some of the methods that Greg was talking about, maybe not time of flight mass spectrometry, but there are other methods for sorting through small particles in the environment that are developed from the engineered nanomaterial world that could be applicable. At least it's heightened our awareness that nano has to be attended to and including in the plastics realm. So the preparedness of the workforce for trying to understand this is number one. And then also, as we have thought about nanoplastics or nanomaterials at large and how they interact with organisms, this maps very well onto plastics, which seem to be, probably less transformable, but we're also able to address that and try to understand using some of the same approaches that were developed through this whole enterprise.
Yeah, I'll just add to that, Elijah, that there's an appreciation in the microplastics community about transformations, about aged plastics being what's out there, and you need to look at that, not just these off-the-shelf plastics. So a lot of the lessons learned from transformations and measurements definitely work for nanoplastics or microplastics in the environment. I mean, they're particles, they behave like particles. A great overlap or a crossover, if you will, is a lot of us, when we were doing nano, would use polystyrene particles for certain studies, right? As a representative nanoparticle. Well, now they're using polystyrene particles as a representative plastic particle. So there is a centering point for all of us to do comparisons on.
Yeah, I'll make one other point about that too, Greg. Thanks. And that is that when Greg was talking about nanoparticles and their transformation in the environment, say with nanosilver and the liberation of silver ions and how that could be a dominant effect over time and in terms of modification affecting organisms, that makes us attentive to plastics in the sense that these are not just solids that sit there. They leach additives, phthalates, and other types of additives and dyes that could be profoundly influential on soil environments, on plants over the long term. And so again, the kind of conditioning to thinking about these issues from the investment of NNI and in nanotechnology research in the environment has prepared us well to be attentive to those issues with nanoplastics too.
Yeah, my 2 cents in here is like, I used to work with PCBs when I was under the DOE working, and so I compare microplastics with PCBs that 30 plus years ago when I was working on them, they were the big issue because they are so stable in the environment. They were not biodegradable. And now we found a way to do that. We found enzymes. So I think to deal with this huge issue of micro nanoplastics is going to be the biochemistry to find how to make these plastic materials biodegradable, because this is a huge problem.
Once again, I'd like to thank our speakers, Greg, Trish, and Jorge, so much for your presentations and for this vibrant discussion. And I'll conclude by reiterating the invitation to follow this webinar series on Twitter at NNI Nano News, and also to check nano.gov for more information. Thanks again for your interest and for joining us here today.
Thank you. Thank you, Elijah. Bye.
Bye, Trish.